Hello everyone, I'm Fan. I'm assistant professor in the University of Colorado at Schultz Medical Campus. Well, computational biology lab doing single-cell multi-omics data modeling using different machine learning and statistical approaches. All right, so we are very excited to present our recent work on a robust computational framework to benchmark spatial temporal trajectory association analysis in single-cell spatial transcriptomic, or ST data. So as we all know that the fast development of spatial transcriptomic technology provide new ways to characterize gene expression patterns along with spatial information, so which offers a unique opportunity to uncover both spatial and temporal information simultaneously. So we wonder how we can best model the spatial transcriptomic data to best characterize the real gene expression variation underlying the data. For example, is that model 1 or model 2 or model 3? Okay, and how the modeling part will influence the biological finding. For example, given a spatial transcriptomic, spatial temporal trajectory or gradient, how we can best identify the gene signatures statistically associated with such component. All right, so given this, we develop a robust computational and statistical benchmarking framework, as you can see here, and we treat the ST count as an input. And first, we remove the low-quality cells and the cell spots with low count. And given a spatial temporal gradient defined by space flow algorithm, we quantify and benchmark 5 different statistical models, which include negative binomial distribution, zero inflated negative binomial distribution, GRM Poisson, zero inflated Poisson, and hurdle. So all this model could be used to model count data, but which one works best for ST data? Then we use a likelihood ratio test to identify the significant genes, which allows us to incorporate other covariates and the technical confounders, like how deep of the sequencing, like NUMI, and other demographic variables like age, sex, or duplicated samples. And in the end, we'll make a decision and suggest which model is the best, and by assessing the goodness of model fit and report the significant signatures. All right, we test our algorithm and framework on 2 typical ST datasets. One is the 10x Vism data from G2 sample breast cancer. As you can see, given a spatial temporal gradient between the tumor and the immune interaction, and we applied and tested the different model and their performance. And in the middle, we presented the model predictions and reflected in the mean and the variation effects. As you can see, these curves are the predicted lines by our model, and looks like the negative binomial distribution and the zero inflated negative binomial are the best to predict the model. Predict the data, and better than the other models like Poisson, right? And furthermore, we use a rotogram to really quantify the model fit at the count level. And in the y-axis, we're showing the squared residual frequency, and as you can see, the line 0 means observed data, and the bars above the line means overestimate, and the bars below the line means underestimate. As you can see that for this particular gene, and both the zero inflated negative binomial and the negative binomial model the count data pretty good and better than the other models. And for the Poisson distribution, as you can see, a lot of overestimate, especially in the low count level, and underestimate in the zero count values. And we get a similar conclusion on the select-seq V2 data from mouse brain hippocampus. So given this spatial temporal gradient in the mouse brain hippocampus, we are able to identify the meaningful genes capture this gradient, as you can see in the middle. And again, the zero inflated negative binomial and the negative binomial really model data very well, which give us meaningful genes that capture this gradient and represent the different parts of the trajectory and the clusters that reflect both the gene spatial variation and spatial aggregation, as you can see in the bottom. Okay, in the end, we want to appreciate our funding partners from Farmer and the T32 grant from MSTP, CU, and Schultz Medical Campus. And if you are interested in our lab, please scan us on the upper right corner, the barcode. And we have several openings for postdocs and computational biologists and bioinformaticians. We welcome everyone to join us and discuss science and do cool computational biology work. Thank you, everyone.